Hi friends, how are you? कैसे हैं आप लोग? Let's start our new topic, molecular microbiology. So in this topic, what we are going to read, we are going to read about different types of PCR. We are going to read about CBNAT machine, which has been used for our TB bacilli. Then we are going to read about MALDI-TOF, which is the very latest machines which are available in a different central institute, and all these are used for the identification of bacteria. So let's start the topic. What we are going to read, we are going to read mostly the amplification. So which amplification we are going to read? We are going to read the amplification of DNA. In this topic, what we will study, we will first amplify the DNA product, and then what we are going to do, we are just amplifying the product, and then we will do the further amplification, and with the help of different machine we will quantify the result. Okay, nowadays the most emphasis is being on quantification. Okay, so this is the basic step of your normal PCR, polymerase chain reaction. So there are the three simple steps which we are being reading from our class 12. So first we will do the denaturation at specific temperature. Then we will do the annealing. Annealing means after denaturation we will use the primer, and the primer will just attach to its complementary site. And then we do the extension will be done with the help of bacterial enzyme called Taq polymerase, and at the same time, at last, we will get the amplified product. You need to understand key two aspect mostly we are using are in the clinical diagnosis. First is the closed system, second is the open system. So first, closed system means key once you put your sample inside the machine, they will quantify the result. Then second method is open method, means you have to put the sample, then they will do the amplification, then again you have to open the machine, take the sample, and you have to use that sample, you have to use that product from the machine to the another machine, and that machine will give you the quantification. So example of the closed system is your gene expert. Then your example of the open system is the mostly PCR which we are using, like real-time PCR. Okay, so nowadays we have both hybrid system, but mostly it is not used in most of the laboratory in India. Okay, so what we are going to read now, we will read the modification of PCR. So the simple PCR is your singlet PCR, means only single DNA, only single target you will achieve or you will diagnose. Now the RT-PCR. RT-PCR is your reverse transcriptase PCR. This is for those virus which has reverse transcriptase enzyme, like your HIV, hepatitis B virus. Okay. So third is your real-time PCR. Means why we are calling them as a real-time PCR? Because in this PCR we are using the fluorescent technology. Then the nested PCR, especially the nested PCR is used for those where we are using two primers. Remember, here we are diagnosing the two target. Now multiplex PCR. Multiplex PCRs means so many targets, so many organisms we are diagnosing in a single go. Okay. Now let's see the mechanism of different PCR. So first is your reverse transcription PCR. Okay. What we are going to do here, first the denaturation will take place. Then the primer will attach to the RNA. First here the RNA will convert into cDNA. Then the cDNA will amplify. So this is the basic difference. Okay. Now second is your real-time PCR. Look at this PCR. The denaturation is taking place. The primer will be there. The primer will have fluorescent compound. Then the primer will attach to its complementary ATCG. Then the amplification, the extension will take place. And after the extension, every step, every extension, every amplification, the fluorescence will release. So that's why we are calling them real time. Means ki fluorescence will detach or the fluorescence will release at each and every step of amplification. So that's why real time. They will give you the graph like structure and they will give you the proper quantification, and this is quite specific. Then your nested PCR. Nested PCR is same like your real-time PCR, but here we are using the two target, means two primer. You are diagnosing two product. So now let me tell you about the specific aspect of this nested PCR. So nested PCR in a normal day, it is mostly used in each and every medical college of India, and it is related to your CBNAT machine. Okay, this CBNAT is your cartridge-based nucleic acid amplification test. This is used for your TB bacilli, tuberculosis bacilli. In this machine, what we are detecting? We are detecting two aspects. First, we are detecting TB bacilli, and at the same time, they will give you the result about rifampicin sensitive or rifampicin resistant organism. Okay, that's why they call it as a nested PCR. Okay, this is the machine. So this machine has a cartridge. We will put the sample into the cartridge and then we will put into the machine and they will give you the result. This is how the inside of the machine looks like. We call them as a gene expert also. Now, the advantage of this nested PCR, first of all, we are detecting the two target. Okay, they have greater sensitivity, they have greater specificity, and at the same time, even if the TB bacilli, tuberculosis bacilli, is present in very few amount in the sputum sample, they can detect. So one thing you need to understand about the limit of detection. So what is this limit of detection means? If we are doing the microscopy, if we are doing the ZN staining, okay, then the limit of detection is 10,000 bacilli per ml of sputum. But here for CBNAT, it is 130 approx. The limit of detection is 130 per 1 ml of sputum sample. Means they have very high sensitivity, more than 1000 sensitivity than the ZN staining. Okay, now the another term, loop mediated isothermal amplification lamp. Here the whole process will be same, but the amplification will take place in the form of loop. So this is the machine related to your lamp. So the mechanism is all same. First denaturation, then primer attachment, then the extension. So the extension will be in the form of loop. What is the benefit of this loop mechanism? Means the sensitivity will increase, the chances of contamination will be less, specificity will be very high. And at the same time, you can detect the positivity or the sample is negative or positive or not with the help of turbidity or fluorescence. You don't need thermocycler machine in this lamp. So mostly nowadays we are using it for malaria, Plasmodium falciparum malaria, and there are other viruses and bacteria also from where we can use it. So this is how it looks like. This is the turbidity they are showing. If the sample is turbid, means that bacteria, virus or parasite is present inside the sample, or you can get with the help of fluorescence also. Okay, this is your lamp related to malaria test, PYVEX. So this is isothermal DNA amplification, and here the target is mitochondrial DNA, and the speed you can get the result within 60 minutes. So for if you are using RT-PCR or real-time PCR, you have to wait for at least 2-3 hours. But for this lamp, the timing will be low. In within 60 minutes, you can get the result. So what is this multiplex nucleic PCR? This multiplex means this is just like your theater. You are going to any multiplex like Wave or any multiplex you are going. There you can watch 3-4 movies at the same time. They are running 3-4-5 movies at the same time. Here the same concept is related to this multiplex nucleic acid test. So same procedure like real-time PCR, but the target is many target we are detecting, more than 2, 3, 4, even 10, 20. So let's get a look of this. This is the machine. This is the BioFire film array machine related to your blood culture. They will give you 20, 30 approx positivity for any bacteria. If the sample is related to any bacteria, they will give it. So if there are different panel for this multiplex PCR. So if the respiratory panel you are using or the machine is related to your respiratory panel, so you can detect these many target. You can see viral target, adenovirus, coronavirus, human metapneumovirus, enterovirus, influenza virus, different types, subtypes, parainfluenza virus. Okay, and in the if you are looking for the bacterial target, Bordetella pertussis, Chlamydophila, Mycoplasma, you can detect in a single go. So this is the blood culture panel. If the gram-positive Enterococcus, if Staphylococcus, Streptococcus is present, they can detect in a single go. Gram-negative Enterobacteriaceae, Neisseria meningitidis, Acinetobacter baumannii, hospital-acquired infection, very common. Candida albicans. Okay. Then now we are moving toward the era of next generation sequencing, NGS. What is the meaning of this next generation sequencing? This is what we are making the address of any bacteria or virus. Why this address is important? This address is important because once what you will do the DNA fragmentation, okay? Then you will make a address according to the ATGC, okay? Then you will make a library and you will protect it for future. And at the same time, you can use that library to making vaccines for that organism. For example, COVID-19, we did NGS. That's why we are getting alpha, omega, delta variants of COVID-19 coronavirus. Okay. So in future, if any virus, any respiratory virus is coming and it is related to your coronavirus, so we can just manipulate or we can just imprint that virus to our library. So we can find, okay, this is related to your delta, this is related to your alpha, these kind of thing. And at the same time, we had used that gene library to make vaccines. So now we will be, if any new virus will come, any new coronavirus will come, we will be like, okay, we have the vaccine and this virus is related to our earlier this or alpha delta variant. So we will be like, okay, no haphazard like COVID waves we have seen in 2020, in 2021, COVID one wave, COVID second wave. Okay, this is the most important mechanism or most important job of your next generation sequencing. Now we are marching toward a term called mass spectrometry. What is this mass spectrometry method? Something will be related to your mass and something will be related to your charge, and then we something it is related to your detection. So let's see. The most common machine used for this mass spectrometry is MALDI-TOF. It is a very highlighted topic. It has been asked in so many UG exam, PG exam, in your INI, NET exam. So what is this MALDI-TOF? So the whole mechanism of this MALDI-TOF is depend upon the full form of this MALDI-TOF. So first is your matrix assisted laser desorption ionization time of flight. So something will be related to the reagent, which we call it as a matrix. Something we are using some laser here. Okay. Then what this laser will do? Laser will ionize the product. Okay. First, the organism will do the desorption. They will take the laser, and then what it will happen? It will leads to the ionization. And then if the ion is charged, whether it is positive, negative, if the ion is charged, they will start moving, because if you are charged, if something is charged, you cannot stop that aspect. They will move. So when they will move, we will detect with the help of detector how they are moving, how fast they are moving. Okay, let's check it out. So this is your MALDI-TOF machine. Okay, so there is a cassette inside your MALDI-TOF machine. Okay, in that cassette, what we are doing, we will put our organism to that cassette. Okay, we will use some matrix. Okay, we will use some matrix. Then what we are doing, we will put a simple, with the help of toothpick. Okay, toothpick, we will take the colony, a very small amount of colony, and then what we are doing, we will just mix with the matrix. Okay, then we will put this plate inside our MALDI-TOF. This is where we are going to put. Okay, then what is going to happen? The machine will release the laser. Okay, and this laser will go inside the cassette. Okay, and what they will do, this laser will ionize the product organism, and after ionization they will start flying, they will start moving. So those organism, those charge, those uh, proteins which are more heavy, they will move slower. Okay, and those proteins which are less in charge, they will move faster. Okay, so if there is any detector, okay, if this is the detector, and from here the this charged particle is moving, so those who are fast, they will touch it quickly, but they are not that much heavy, so they will come first, but the impact will be less. Okay, and those who have more charge, they will move slow, but when they will just touch it, they have a more powerful impact. So this flight, we will record it, and then the impact we are recording. So after that, what we are getting the graph. Okay, like this, these smaller one will be, they are coming quick, to have less charge, they will come first and make a lesser impact, so the graph will be small. Then again, those who are coming quite late but they have more charge than the earlier, they will make a big graph. Then those who are coming late but the charge is very high, so they will make a bigger graph. So that's how we will make a mass spectrometry. Okay, and then we will make a database. So whenever we are going to detect the new organism, we will match that organism proteins with the charge, mass by charge ratio, with our database. So if that organism is showing like your Escherichia coli, so we will diagnose with the help of diagnose it as a Escherichia coli. If that organism is showing like Pseudomonas, making a graph like Pseudomonas, so we will diagnose them as a Pseudomonas. So the time of detection of all these is only 20 minutes. If we are using a normal procedure, if we are using our traditional method, it will take so many days, 2-3 days at least, for the identification. But if we are using this, if we are getting the colony within 20 minutes, we can diagnose what is our organism is. So it is very helpful for our AICU patient, ICU patient, like adult ICU, pediatric, neonatal ICUs. Okay, this is how the whole procedure looks like. So this is the graph related to your MALDI-TOF. So first we will do these mixing of matrix with the sample, then the laser will come, then the ion will release in the presence of electric field, then we have a detector, they will just make an impact to the detector, the different this mass spectrometry will form, and this is how we go. So that's all for your topic. So stay blessed. Bye bye. Like and subscribe. Bye bye and take care.